this side.
Yeah, I'm taking this.
Okay.
This is not like taking any action. No? Just a. Yes.
So again, you can again type share screen, and if it's not working, then that's answer. We will go like this.
Hello.
Hello.
Can you hear me?
Yes, sir. I am showing. Yes, sir.
Are they both showing?
No, sir. Full screen mode is not showing.
I'm trying to do it on full
So
screen.
that's fine. If we can go like this, sir. If it is not working, then it's
Just give me a minute. I will try again.
sure, sir. Take it. No, sir.
लाइटिंग ज़्यादा नहीं बढ़ रही, लाइटिंग दिखाई नहीं दे रही। लाइट ऑन करके देखिए।
PCI relating and is affected to HPT or Mirpur. The aim is to provide the growth and development of pharmacy education in the state of Himachal Pradesh. So we are organizing this international conference dedicated to highlighting recent scientific and technological innovation that contribute in drug discovery, formulation development, molecular biology, pharmacogenomics, clinical research, and life sciences with a focus on emerging tools, novel therapeutic strategies, and integrated approaches. So now let me introduce you our eminent speaker, Dr. Sachin Goyal. So Dr. Sachin Goyal is currently working as a staff scientist in the Department of Anesthesia Research at Cincinnati Children's Hospital Medical Center, Cincinnati, Ohio, USA. He obtained his PhD in pharmacology from R&T Government Medical College, Udaipur, Rajasthan, India. His primary area of research is neurobiology of pain, where he employs diverse experimental approaches such as development of acute and chronic pain models, assessment of reflexive and non-reflexive pain related measures, in vitro and ex vivo whole cell patch and electrophysiology of cultured non-dorsal ganglion, trigeminal ganglion, spinal cord and brain slices along with cultured human dorsal root ganglion neurons. In addition, his work involves advanced imaging techniques and gene expression studies to elucidate brain associated pathways both at peripheral and central neuronal levels. These laboratories projects are supported by funding from NIS, the Department of Defense.
Okay,
क्या करोगे सर?
good morning to all of you.
घर लगा
I
दिया
am
सर,
Sachin
चलो।
Goyal. Like Shivan given me, has given me my introduction. Today's my presentation is based on the neuronal electrophysiology and effective tool for pain research. So all my career was based on the pain research in India and abroad also. So today I want to like deliver a small talk, like brief talk on neuronal electrophysiology, such a very wonderful tool for neuronal, like basically neuronal physiology, electrophysiology. So this is like our setup, like Cincinnati Children's Hospital. This whole campus for Cincinnati Children's Hospital is situated in the Cincinnati. The Cincinnati Children's Hospital comes among the like top 5 pediatric hospitals across the world. Like so many innovative research has been taken place in the Cincinnati Children's Hospital. Very well known like polio vaccine, oral polio vaccine discovered in the Cincinnati Children's Hospital. As you know, the oral polio vaccine discovered by Dr. Savin, and Dr. Savin was the researcher, scientist in the Cincinnati Children's Hospital, and he discovered the oral polio vaccine. So very good contribution of Cincinnati Children's Hospital in the medical field. So first I would like to talk about the pain just a little bit. We all know about the pain. Pain is the emotional and sensory experience associated with the actual or potential tissue damage, or can be described in terms of such damage. So pain is like the initial signal that something is going wrong in the body. It may be injury, it may be like something wrong is going in the visceral organs of the body. So pain is the initial symptoms that we have to like take care that something is going wrong in our body. So pain can be divided into two forms, acute pain and chronic pain. The acute pain can be defined as a pain, less lasting pain. Means like pain that occur like not more than 2 months. That is acute pain. The chronic pain that the long lasting pain, more than 2 months, like the pain occur, that is like chronic pain. So chronic pain further can be divided in 3 different categories: psychogenic pain, inflammatory pain, and neuropathic pain. Neuropathic pain basically the nerve injury induced pain. and further divided peripheral pain and central pain based on the the level of the nervous system. So peripheral is like all of you are, I think, like pharmacy student, and all you study the anatomy. So the peripheral is like the peripheral axis of the body. The central is like central axis of the body which contains the spinal cord and brain. So on the basis of the site of injury, we can divide the neuropathic pain in peripheral as well as central. Then it's just like a brief discussion about the pain or like pain physiology, how any person like can feel the pain. So something like going on in the nervous system. That's why finally your brain identify the signals as a pain. So when any injury, tissue injury take place in any part of the body, then the receptors which present on the neurons, that will activate. That will activate because of the pressure or variation in the temperature or chemical means. When the receptor activated, they generate the action potential. Action potential means they convert the pain signals in the electrical signal, because the neuron, the nerve which is present in our body is like the electric wire. So the information regarding the pain it will convert in the electric signal on the neuron, nerve, and that electric signal then continue going towards the higher sector. That means pain. Ultimately, the feeling of pain is out of pain. So those signals in anywhere in the body, anywhere like the injury take place, the electric signal will generate because of the activation of the receptor, and that electric signal goes towards the pain via the spinal. So the first you can like see the peripheral side, if the electric signal generated, they will like go towards the spinal cord through the DRG, dorsal to ganglia. This is the relaying center. And from the spinal cord, the sensory nerve goes to the brain, and then those signals analyze, and finally they will like identify as the pain. And the brain take the decision how we have to like reduce or minimize the cause of the pain, and then the signals we generate in the brain will propagate through the motor neurons at the affected site. So this is the like general physiology, general mechanism of pain signal generation and identification of the pain. Now comes on the main topic. If any person is like doing the pain research, what the tools available to do any kind of pain research, like preclinical pain research or clinical pain research. So according to my lab, actually the work carried out in my lab, we are using the animal or human models of the pain, specific pain condition, behavioral assessment tools, how we can assess the pain symptoms in animal as well as the human. Molecular and biochemical techniques we use frequently to identify what molecular and cellular changes taken place in the body after the pain condition occurs. And the fourth one is like neuronal electrophysiology. This is very important. Neuronal electrophysiology. By using the neuronal electrophysiology, we can like access the excitability of the neurons. Very exciting technology. And in the later of my presentation, I will like show you what the importance of this technology. So these are some, I'm showing here some animal pain models like CCI, chronic constriction injury model, spinal nervalization model, partial sharing nervalization model. The selection of the animal model depends on which kind of pain condition you are like, taking for your research. These are the spinal cord slices, immunohistochemistry slices, so we can like access the expression of the different gene level or different target level at the different level in the spinal cord. And these are the DRG, dorsal root ganglia immunohistochemistry. So by these techniques like molecular and cellular techniques, we can like identify the level of expression, either the like expression of the gene or protein or any specific marker is going to up-regulated or down-regulated after the pain condition. So we can like use these techniques. But today's my presentation is focused on the neuronal electrophysiology. So just let's talk brief in the neuronal electrophysiology. So in my lab, the neuronal electrophysiology, we are using the different tissues from the nervous system. Like for example, dorsal root ganglia from mice as well as human donors, trigeminal ganglion, which present in the skull, spinal cord slices to identify, to access the excitability of the spinal cord neurons at the different levels. Periaqueductal gray slices in the brain, and cell lines, different cell lines. For example, human-induced pluripotent stem cells, lion cell lines we are using, head cell lines we are using. So, actually, the research should be in the translational nature. The today's like scenario that demands your research should not be like based only from the animals. That research should be go from animal to humans. So the nature of the research should be translational in nature. So initially we start from the mice, and then the data of mice which we receive, that we translate in the humans also. So the nature is translational type of research. This is the human spinal cord. We extracted, we regularly receive the human donor spinal cord samples from the human donors. This is the live spinal cord. The neurons and the tissue present in this spinal cord is live, not dead. So initially, like when any dead body or something like comes in our hospital, because we are working in the hospital, so we initially from the donors, human donors, we extracted the spinal cord. So this is whole spinal cord. These are the spinal nerves, and here, if you can see, these are the dorsal root ganglia. So this is the relay center basically. They regulate the frequency and firing of the neurons. So we extracted the spinal cord and then extracted the DRG and cultured the DRG in the artificial media, in vitro culture, and then those in vitro cultures, like these are the culture the neurons will be dissociated, separated. So these are the single neuron. These are the single human DRG neurons. This is the neuron which present in your body, in my body. This is the neuronal cell. So we patch. Later on I will show you how we patch and what the importance of this technique. So we patch this and provide the current to the neuronal cell. And because of the current stimulation, the cell will get activated at some point, and we get the action potential firing and excitability parameters that we will record. So these are the different tissue level and cell level which we are using for neuronal electrophysiology. This is the setup of neuronal electrophysiology in my lab. This is the you can we said it rig, the electrophysiology rig. This is the whole setup. This is the rig. This is the software and different connectors connected to the rig. And this rig is this is the very sensitive microscope. This is the fluorescence microscope. So we can see the single cell under the microscope, and this cell can be imaged on our screen here. I will show you later on. And to provide the, because we need the live cell during the recording, electrophysiological recordings. So we provide the own artificial media which present in our body, and all the gaseous exchange we continuously provide. So these are the oxygen and carbon dioxide cylinder, carboxygenate, the we can say, and the artificial fluid exactly same as the human fluid. So that artificial fluid we provide, so cell can be live longer, and we can record. So this is the basic setup, very sensitive and very expensive setup this is. So we use this technique. So this is the recording, you can see when our cell is present in the rig. This is the cell, you can see here by the page. This is the capillary, which capillary contain the electrode. And when the capillary touch the cell, the electrode come in the contact with the cell, and we provide the current, electric current to the cell. Start from the picoampere. Picoampere is very small amount of the current which we provide to the, which we passes through the cell membrane. So we start from the 0 picoampere, then -10, -20, -30, -40, 10 picoampere. 10 picoampere increment we provide the current to the cell up to the 200 and 300 picoampere. So at the certain point, I think like you know the threshold. The threshold means at the point when anything starts to act. So at any point, some point, when we start the current passing through the cell from minus 10 picoampere and start with the increase with the 10 picoampere increment, then minus 10, minus 20, minus 30, minus 40, something like that. So that will go 0 and then 10 picoampere, 20 picoampere, 30 picoampere, 40 picoampere, and at the some point the cell gets the sufficient current to stimulate, and suddenly like cell start to fire, like just start to create the action potential, which means the stimulus, current stimulus convert in the signal, electric signal. So that's the neuron. Neuron is like working as an electric wire. So our information, or we can say the noxious information in the form of the current, electric current, they convert in the threshold. At the point of threshold, they convert in the action potential. And that action potential goes like towards the neuron, one neuron to second neuron, adjoining neuron, and ultimately reach out the brain, where that signal read or analyze as a pain condition. So this is all our setup. I will like show you how we record and what the action potential is. So I want to like show you here, like in my earlier slide, I show you we page the cell. Like now my electrode is in contact with the cell, and now we start to inject the current with the 10 picoampere increment. So when we inject the current, and this kind of like you can like see here, we injected the current to 10 picoampere increment, and suddenly cell start, when the threshold will come for that cell, the suddenly like start to fire. This is the point where the cell gets sufficiently activated. This is the threshold, the minimum force required to excite, stimulate the cell to produce the action potential. So that current information is converted in the signal. This is the action potential. Further we can like see, this is the action potential. I think like in the physiology, you read this, that the threshold for the human cell is minus 90 millivolt. Some approximate minus 90 millivolt. So if anyone, for example, just एक आपको simple language में एक example दूँगी, कोई आपको पेन टच करता है आपके हाथ पर, तो जब वो हाथ पर आपके पेन टच करेगा, तो जो स्टिमुलाई है, नॉक्सियस स्टिमुलाई, पेन टच कर रहा है तो स्टिमुलाई दे रहा है, फील हो रहा है, लेकिन वो फील क्यों हो रहा है? At some time, in some point of pressure, that information convert in the action potential, electric signal, this kind of electric signal, and that electric signal goes towards the brain through the spinal cord, and ultimately brain discover that there is a pain signal, and also analyze at which part of the body the pain is like generated, so that brain can be like take the corrective measures to reduce or eliminate that cause of the pain. So this is the electric signal. So we record each kind of electric signal and so many parameters from the different kind of the neuronal cells and tissues, and further analyze. After analysis, analysis of the various properties, neuronal properties, excitability properties, for example, intrinsic properties, resting membrane potential, we can like calculate, analyze by using different softwares, and Rio base minimum current required to decide the firing, that is Rio base, and firing frequency, the firing frequency and like action potential for latency of the action potential. So many parameters like we can like analyze by using these recordings, by using the different softwares. So finally, these kind of recordings we are like doing in our lab. And these are the, after like recording, like we analyze using the data, and these are action potential waveform. This is the signal, electric signal which goes towards our brain. These are the RMP firing. I don't know how much you are understanding this technique, but I am trying to like give you the brief understanding of the electrophysiology and nature of the neuron and intrinsic properties of the neurons. And this is spontaneous firing. Spontaneous firing means like neuron fire without any stimulus. This is the spontaneous one. Just Q, spontaneous. So these kind of like recordings we are like doing in the neuronal electrophysiology on different human cells, human tissues, as well as on the animal models, animal cells. And these like over these my work in my lab, like that is has been like published in recently published, like these are all lab recent publications based on the neuronal electrophysiology. So this is like recently published articles on different pain conditions. Like this is like we are like working to develop a humanized P2X4 SCOB. P2X4 is the receptor, and we are like trying to develop a single chain fragment variable antibody. So if we develop this kind of antibody, then only one tablet capsule or like one injection will be sufficient to relieve from the pain condition for long, for months. Only one injection or one capsule, whichever the formulation will be, that will be effective, or that will be sufficient to prevent the pain in chronic pain condition for months. This is the visceral pain related acute pain condition. This is the SCLP. We are like working on that to develop the formulation for this monoclonal single chain fragment variable antibodies. So all these are like publications which are related with these electrophysiological techniques. And this is my group. We are like working together in our lab for neuronal electrophysiology. Some of the best brain in the world which are like working in the neurobiology of pain condition. So basically, just I want to like show you what kind of like work regarding the neuronal electrophysiology is going place in the world. And this electrophysiology, very few things are like working on this, and specifically on the translational research. So I believe that from some of you students like in future like try to like or want to like work in the pain research or like neuronal physiology, electrophysiology, and this brief knowledge like I think like motivate some of the students, I suppose, and I believe. So this was my presentation for today, just brief presentation. If any question you have, then just like ask me.
Thank you, sir, for this great lecture. We have few questions from you from the student sides who are watching you online. So first question is, how can we utilize this technology in case of neurodegenerative disease? So sir, you have clearly elaborated about neuropathic pain. But can we utilize this technology in case of neurodegenerative disease such as Alzheimer's or Parkinson?
Yeah. Like neurodegenerative disease, like for example Parkinson. What is going on in the Parkinson? That the neurons which present in the like central nervous system, they are like get activated, more activated, or we can say the hyperexcitable condition. So we can like check, or like we can compare the naive condition with the disease condition. So what the property of neurons like going to change from the normal human, normal live animal to the diseased animal. So because the firing frequency is going to rise in the Parkinson and like you can say any kind of Alzheimer's or Parkinson, the patient is like continuously, the brain is receiving, or the cortex portion of the brain is continuously receiving the firing. Action potential are going to, if the normal speed of action potential generation is like this, in the Parkinson and Alzheimer or any kind of neurodegenerative disease, the firing frequency goes like this. So more signals are like coming in the brain, and the brain is like confused. And that is like hyperexcitable condition of neurons in the brain. So we can like check the electrophysiological properties of neurons by using this technique in the Alzheimer and Parkinson. And correlate the animal study with the human studies, human samples.
Thank you, sir, for your explanation. So next question is, can the integration of artificial intelligence is possible in this technology?
Yeah, we are using, but not so much extent. Like we are using just coding, because when we do the electrophysiology, the large data set we receive, very large data set, because we record the properties as I showed you in my presentation, and then we have to use the different software to analyze those data to conclude the records. But if we are doing manually, the one by one we have to take one by one recordings and then analyze manually by using software. But if we have the code, so we can like take so many recordings at a time, same time, and just using the code we can like analyze within a minute if you have the perfect code. So the machine learning and artificial intelligence can help us like so much, and we are like using those things. Artificial intelligence, specifically machine learning, we are using very much.
So it's all dependent on the algorithm, sir. Thank you for your clear explanation, and thank you, sir, for this great lecture. This whole session was very informative. You have shared in detail about neuropathic pain, neuronal electrophysiology, electric threshold, which can stimulate the neuronal cell. It must have imparted a lot of knowledge in the mind of students present here, watching you on YouTube live. So I think this is the end of the session. Again, sir, thank you so much for being a part of this event. But before that, I would like you to accept this as a gesture of appreciation, sir, please.
Thank you. Thank you so much. Thank you.
Thank you again, sir. It was very informative session, and thank you for being a part of this. Thank you.
Thank you, and good luck to all of you. Thank you.